Hello everyone, I'm Roaa Alayoubi, and in this video I'm going to be talking about phototransduction. The photoreceptor layer is found just above the pigment epithelium, and the photoreceptors are divided into three segments: an outer segment which is closest to the pigment epithelium, an inner segment, and the synaptic body which synapses with different cells, mainly the bipolar and the horizontal cells. The outer segment of the photoreceptor has sodium channels, and the inner segment has sodium-potassium pump and potassium channels. Just as a recap, we know when light hits the retina, it causes the conformational change of cis-retinal into the trans-retinal form, and then opsin, which is found in both rods and cones, activates the G protein transducin, which in turn activates PDE, phosphodiesterase, which causes the breakdown of cyclic GMP into 5-GMP. Now, why is cyclic GMP important? As you can see over here, it is bound to the sodium channel, which allows sodium influx into the cell. Now, when this is inhibited by PDE, there will be less sodium influx into the photoreceptor, and it will be hyperpolarized, yani more negative. This will cause less calcium influx at the synaptic terminal, and ultimately less glutamate at the synapse between the photoreceptor and the bipolar cell. So light causes the photoreceptor to be hyperpolarized and ultimately less excitatory neurotransmitter at the synapse. So in the darkness, it's going to be the opposite of this. The photoreceptor will be depolarized and there will be more glutamate in the synapse. So the question is, how will light, our stimulus, then eventually cause an action potential to be generated if it causes hyperpolarization and less neurotransmitter? The answer is in the type of receptor which glutamates act on. We're going to talk about two glutamate receptors, the AMPA, which is excitatory, and the metabotropic, which is inhibitory, metabotropic glutamate receptor. The AMPA receptor is also an ionotropic receptor. You can remember that AMPA amps up the cell, therefore it's excitatory, found on the off-center bipolar cells, and metabotropic is inhibitory, found on the on-center bipolar cells. Horizontal cells, which are found in the inner nuclear layer, are inhibitory and release the inhibitory neurotransmitter GABA. So we said by the effect of glutamate, the off-center bipolar cell will be depolarized and the on-center with metabotropic receptor will be hyperpolarized. When do we have high glutamate? In the darkness. So this is a photoreceptor in the dark. As you can see, it is depolarized, having more glutamate at the synaptic terminal. Which cell will be depolarized? The off-center cell, which is excitatory when we have glutamate, and the bipolar cell will be hyperpolarized with the one with the metabotropic receptor. Now in the light, the photoreceptor, as we mentioned before, will be hyperpolarized, having less glutamate at the synaptic terminal. So the off-center cell, which was depolarized by the effect of glutamate, in cases of less glutamate, it will be hyperpolarized, and the on-center cell, which was inhibited by glutamate, will be depolarized in the light because we have less glutamate. The surround of the receptive field also influences how much glutamate is released into the synaptic terminal. So if the surround is dark, the photoreceptor found there will be depolarized just as usual, having more glutamate at the synaptic terminal between the photoreceptor and the horizontal cell, which we already discussed, which is inhibitory. So having more glutamate at the synaptic terminal between bipolar and horizontal cell causes the horizontal cell to release more GABA, more of the inhibitory neurotransmitter, causing even less glutamate at the synapse and the depolarization of the on-center bipolar cell. Now when the surround has light, the photoreceptor will be hyperpolarized, there will be less glutamate between the photoreceptor and the horizontal cell, so there will be less excitation of the horizontal cell, the inhibitory cell, it will be hyperpolarized, and there will be no GABA release, so there will be less or no inhibition at the photoreceptor, which is at the center, and there will be more glutamate release, causing the depolarization of the off-center cell. So shortly, in light, the photoreceptor will be hyperpolarized, the on-center cell will be depolarized, and in the dark, the photoreceptor will be depolarized, and the depolarized bipolar cell is the off-center cell. And just like how we have off-center and on-center bipolar cells, we have corresponding, often on-center ganglion cells, which will generate an action potential and send it through the optic nerve to be processed in the visual cortex.